Welcome to this deep dive. We're sifting through some recent sources, pulling out key updates in cancer research.
Exactly. We'll be looking at, results from ongoing clinical trials, but also some really exciting new discoveries from the lab.
Things that could shape future treatments down the line. Our mission really is to give you a clear picture of what's new and, importantly, what it might mean.
Yeah, and what's interesting is how it covers the whole spectrum.
Yeah.
We've got studies that could impact care relatively soon, and then there's more fundamental stuff opening new doors.
Because let's start with the clinic. Source 1 details the AMPLITUDE trial. That's AMPLITUDE.
Right, it's a phase 3 study, NCT 0497844, big trial.
And it looked at metastatic castration-sensitive prostate cancer.
Specifically, it tested combining Niraparib, that's a PARP1/2 inhibitor, with abiraterone acetate and prednisone.
Okay, and the focus was on patients with HRR gene alterations, like BRCA1 or BRCA2.
Precisely. HRR is key for DNA repair. So if it's faulty, like with those BRCA mutations, the cancer cells are, well, more vulnerable. That's where PARP inhibitors come in, targeting that same vulnerability.
And the results, I gather they were pretty significant.
Oh, absolutely. The combination showed a significant improvement in radiographic progression-free survival, basically how long before the cancer visibly grew on scans.
And the difference was big.
Yeah, the median time wasn't even reached in the combination group. Compare that to 29.5 months in the control group. That translates to a 37% reduction in the risk of progression or death.
37%, wow.
And crucially, the benefit was even stronger in that specific subgroup with BRCA1 or BRCA2 mutations. Really stood out there.
Right. And did it also delay symptoms?
It did significantly delay the time to symptomatic progression, again particularly noticeable in the BRCA subgroup.
What about overall survival? Still early.
Still being collected, yeah, but the early trends look favorable. Promising signs.
In side effects, combining drugs often means more toxicity.
It did. Grade 3 or 4th adverse events were more common, 75.2% versus 58.9%. Mostly anemia and hypertension.
But manageable.
Seems so. Treatment discontinuations due to side effects were relatively low, so, suggesting it was generally tolerable for patients. The key takeaway here really is this combo might set a new standard for this specific group of patients.
Okay, interesting. Let's move to source 2. This is about a new drug entering trials, ACE-232.
Yes, ACE-232. It's a novel oral CYP11A1 inhibitor.
CYP11A1. Okay.
And the first patient just got dosed in a phase 1 trial. That's NCT 068012336. This is for metastatic castration-resistant prostate cancer, or mCRPC.
Phase 1, so early days. Safety, tolerability, finding the right dose.
Exactly. It's a two-part trial. Dose escalation, then dose optimization. Looking for safety signals and any early hints of efficacy to figure out the dose for phase 2.
And the thinking behind this target, CYP11A1.
Well, preclinical data looked good. It suggested ACE-232 might have a better profile than some earlier inhibitors. The goal is to suppress androgen production, a key driver for prostate cancer, even when it becomes resistant to other hormone therapies.
So another way to cut off the fuel supply.
Basically, yes. Trying to block that growth signal more effectively.
Got it. And source 3 brings in another new agent, XL309.
Right, XL309. This is a USP1 inhibitor, also in phase 1 trials.
USP1 related to DNA repair again.
Yes, like PARP USP1 plays a role there. So this trial is testing XL309 by itself and combined with Olaparib, the well-known PARP inhibitor.
And this is also for patients with specific mutations.
Yeah, advanced solid tumors with BRCA1 or BRCA2 mutations, and that includes mCRPC patients.
So hitting a similar vulnerability pathway but with a different tool, potentially alongside a PARP inhibitor.
That's the idea. Preclinical studies showed, robust activity, especially in those BRCA mutant tumors. And importantly, they suggested synergy with PARP inhibitors.
Synergy meaning the combination works better than either drug alone?
Exactly. So the insight is targeting USP1 might boost the effect of PARP drugs or maybe even help overcome resistance when it develops. Lots of potential there.
Okay, so quite a bit happening in the clinical space. Let's pivot now, as you said, to some of the preclinical work. Source 4 looks at something familiar, aspirin.
Yeah, aspirin in cancer. It's a topic that keeps coming back. This recent research suggests maybe aspirin helps stop cancer spread, metastasis, by boosting the immune system.
Boosting the immune system, how?
So the thinking is it blocks an enzyme called COX-1 in platelets. that stops platelets from releasing a molecule called TXA2.
Okay, TXA2. Why does blocking that matter?
Because TXA2 seems to weaken T cells. Those are the immune cells that should be attacking spreading cancer cells.
Ah, so TXA2 puts the brakes on the T cells.
Sort of, yeah. So if you block TXA2 with aspirin, the T cells stay stronger, they multiply better, and they can do their job fighting the cancer cells.
And there's evidence for this.
Well, it reduced cancer spread in mice studies. And looking at human data, there seems to be a correlation. Aspirin users sometimes have a lower risk of metastasis and fewer cancer deaths in early-stage disease.
Interesting. So it's not just about blood thinning.
Potentially not just that. This immune mechanism could be relevant for prostate cancer, too. Researchers are now thinking maybe we can develop more specific TXA2 inhibitors to get this benefit without aspirin's other effects, like bleeding risk.
A new angle on an old drug. Fascinating. Now, source 5 takes us somewhere completely different, zebrafish embryos.
Right, zebrafish. Researchers found these tiny molecules called MicroRNA as in the embryos, specifically one called MIR2-185P.
MIR2-185P, and what does it do?
It seems to act like a brake on something called the PI3K pathway.
PI3K. That pops up a lot in cancer, drives growth and spread.
In many cancers, yes, including prostate cancer. So in the lab, extracts from these embryos containing these microRNA actually changed how cancer cells behaved.
Change them how?
They moved less, were less invasive, and they started sticking together more, kind of like normal cells do. They also saw changes in key proteins, boosting P53, which is good.
A tumor suppressor.
Exactly. And lowering another one called TCTP, which is linked to growth.
And zebrafish share genes with humans, right? So the idea
is still very early, is maybe we can use these natural molecules to sort of reprogram cancer cells by targeting these key pathways. A totally novel approach.
Very different. Okay, finally, source 6. This touches on artificial intelligence, AI.
Yeah, a major leap in protein design using AI. Now, this isn't a cancer drug itself, not yet anyway.
But it's a fundamental breakthrough.
Absolutely. Researchers have engineered artificial proteins that can actually move and change shape dynamically. That was something only natural proteins could do before.
Okay, dynamic artificial proteins. What are the implications for cancer?
Huge potential. The key insight is we might be able to design smart proteins. Imagine proteins acting as highly sensitive biosensors to catch cancer markers super early.
Or delivery vehicles.
Exactly. Proteins designed to deliver drugs only to tumor cells, sparing healthy tissue. Or maybe proteins engineered to specifically block the interactions that drive cancer growth.
Precision medicine at a whole new level.
Potentially, yes. It opens up a whole toolbox for designing custom biological tools.
So recapping today, we've seen quite a range from phase 3 results like AMPLITUDE that could change practice soon for some,
to brand new drugs like ACE232 and XL309 just starting human tests.
and then these really fundamental discoveries: aspirin's potential immune effect, molecules from zebrafish, AI designing moving proteins.
It really underscores how fighting cancer happens on so many fronts. You need the steady progress in the clinic, testing combinations, refining treatments.
But you also need these, well, almost revolutionary ideas coming out of basic science labs.
Definitely. And it leaves you thinking, doesn't it? How might these different threads, a molecule from a fish, an AI protein, a new look at aspirin, how might they eventually connect?
Could they converge to create entirely new ways to tackle cancer, combining different approaches, maybe?
It's certainly provocative. We might see completely novel strategies emerge by integrating these different lines of research down the road. It's definitely an exciting and hopeful space to watch.
That's all for today. Stay strong and fight on.